In the last part of Chapter 4, Part 2, I want to talk about a couple more things. One of them being another type of layer bacteria secrete around themselves, another type of glycocalyx layer called the capsule. So if we jump back to Chapter 4 again to connect where we mentioned this last time, Chapter 4, Part 1, that is, we talked about a capsule as being one of the two types of glycocalyxes, a slime layer and a capsule. One of the significant aspects of a capsule is the extra protection it gives to the bacteria that secretes it. So here you'd be looking at the bacterial cell itself. This is like, for example, here, the cell membrane, and here is the cell wall. So we'd be looking at like a gram-positive bacteria. So then the capsule will be secreted around that. So it would enclose that whole thing. In fact, if we just go back real quick to this figure we've been showing over and over in 4 Part 1 here, this shows the capsule here as this tan layer on the very outside here. In fact, it points to it right here. So that's the capsule. So it's a layer of protection. It does a variety of things. It can help bacteria stick to surfaces in addition to just kind of helping them survive different exposures in different types of environments. The capsule can help with water absorption, water retention. It can help with the microbes sticking to different surfaces. So there's a variety of little nuances there. From a medical standpoint, and again, I like to try and keep it in that reference, from a medical standpoint, they actually have an even more significant aspect to them, and that is that they help them avoid the immune response that all of our bodies would typically go into when exposed to these types of bacteria. And we're putting the cart in front of the horse here a little bit, because we're actually going to talk about the immune system in more detail later, but we haven't done that yet. So later we'll understand this better. But today we're going to introduce a concept that shows you kind of how the capsule helps protect the bacteria, and also a little bit about how your immune system works. The way the capsule protects the bacteria is it protects them from a process in the body known as phagocytosis, and this is a process carried out by phagocytes. So they prevent, the capsule will prevent a bacteria from being destroyed by cells in the body called phagocytes, and these are a type of cell of the immune system called a white blood cell. Most of you are probably familiar with white blood cells, what that generally means, part of your immune cells. One of the general classifications of a white blood cell is called a phagocyte. A phagocyte is a cell that literally eats other cells. This is actually a literal thing. So the cell will literally engulf other cells, bring them inside, and digest them. I've got a video and some figures I'm going to show you here in just a second, so this will all make a little more sense visually. But we're talking about your immune system having cells that will eat other cells, and we're talking about a capsule here on a bacteria that would normally be eaten. The bacteria is normally the food for your white blood cells, but the capsule is like this armor that protects it from that process. So when a capsule then is found on a bacteria, what that means is it's harder for your immune system to remove them, and you can see then how that might be bad, because your immune system needs to remove these cells to keep you healthy. Now, it's not a huge problem, but it's something that contributes to bacteria being a little bit more pathogenic, a little bit harder to get rid of by the body. Now, from the notes here, I want you to draw this out. I'm going to show you a few figures first, and then we'll draw it together here. And then we'll answer this question. So you can hold off on that for just a second. Here's a picture of the capsule. Here we go. I pulled up the wrong figure here. Get to it here in a second. Here's a picture of a capsule around a bacteria. And I thought I had a picture. Let's do this real quick. Here we go. So here's what, tell you what, let's just pull up phagocytosis first. I do have a video. I'm going to show you that next here. Here we go. Here's what phagocytosis generally looks like. Here's a white blood cell. Not much detail given to it, but what this white blood cell will do is literally attach to a microbe, like a bacteria, and it will literally swallow that bacteria and enclose it in a vesicle inside the cell. And inside that vesicle, the cell is releasing digestive enzymes into the vesicle with the bacteria, And together, the digestive enzymes will chop the bacteria up and kill it, effectively neutralizing that particular bacteria. And then the phagocyte literally spits out the remains on the back end there. Here's a better picture showing the same process. So here's your microbe being swallowed, being converted or being infused rather with digestive enzymes, those breaking down the bacteria and the parts being released there. So that's kind of the normal process of phagocytosis. What happens in a lot of cases with the capsule is that the capsule will basically prevent that bacterium from being fully engulfed by the phagocyte. And the capsule basically acts as a repellent to the phagocyte in their ability to capture and grab a hold of that particular bacteria. So you can almost think of it like a repellent for the of the bacteria to the phagocyte. So when a bacteria has that, then they're going to be harder for the immune system to get rid of. Now let's go back here then and draw that three-part process. So we're going to draw a phagocyte, and we're going to draw the phagocyte engulfing a bacteria here. So in step one, we're going to show the phagocyte, and then we're going to show bacteria right next to it. There's your, let me make that a little thicker. Here's your phagocyte. It's kind of an irregular shape to it. And then next to it, you've got a bacteria, much smaller. And then we're going to show the phagocyte attaching to the bacteria. I have to use our imagination quite a bit here. So here's our phagocyte, here's our bacteria, and we're showing the phagocyte attaching to that bacteria right here at that attachment point. Okay, so that's step one. In step two, we're going to show the bacteria being partially engulfed by the phagocyte. And what's happening here, and we're still showing the attachment sites. This is where the phagocyte is gripping a hold of the bacteria. So what's happening is that the cell is literally moving and malleating itself. I think I just made that word up. It's flexing itself around the bacteria, and literally like a mouth, almost opening up out of nowhere. It kind of begins to swallow this bacteria. So that's step two there. And in step three, we're going to show that the bacteria is now trapped inside of a vesicle. This little vesicle forms when the bacteria is brought in fully into the cell. And we're going to then show that the bacteria is killed by simply just drawing an X over it. And what that means is that the bacteria then inside that vesicle is now killed by digestive enzymes. We're actually going to learn about these steps later in the semester in a little more detail, and we're going to kind of name and classify each step, and I'll expect you to know them a little better then. So for now, we're just going to keep it to this kind of simple thing. Phagocytosis is attaching to the bacteria, drawing it inside, and then enclosing it in a vesicle where it's killed. That's basically what phagocytosis is. Now, oops, wrong one. Now the next question rather was, which step of the phagocytic process do capsules inhibit? And generally speaking, and the answer that I want you to know here, in most cases, there's always a few exceptions here, but in most cases that process is the attachment. In other words, phagocytosis, let me just spell it out here. Phagocytosis, I said that wrong, excuse me. Bacterial capsules prevent attachment of the phagocyte to the bacterium. So the key part there is they prevent attachment. Step one. All right. So the phagocyte can't really ever grab a hold of it. Really what we would see here, if we want to make a slight variation of this off to the side, if we do a variation of this over here, what we would see is that capsule would prevent those arms from being able to grab onto it. Use the word arm in air quotes there. So there's the attachment sites. Your bacteria, here's the bacteria. The bacteria is not going to have a capsule around it. And so the ability of those attachment sites, they're actually called receptors, the ability of them to actually grab a hold is no longer good enough to make that grip and that attachment. All right. Now, the other key part about this is that what this really does is it gives the bacteria a little bit of a leg up in their ability to avoid the immune system. It doesn't mean that they can't be killed by the immune system, because they certainly can. There's other ways the body's immune system can attack them. So it doesn't give them a free pass and make them totally immune to your immune system. But what it does do is it makes them just a little more dangerous. It gives them one way to get around one part of the immune system. So in certain circumstances, that capsule can give them the edge that they may need when it comes to causing infection. And so in some cases it's significant, in other cases it's less significant. But the idea is that it gives them an edge. So if you have a bacteria that's already a pathogen, say it's already got a harmful nature to it, and then it has a capsule on top of that, then usually it's that kind of combination that makes bacteria worse. The ability to be a pathogen and have a capsule, making it harder for the immune system to get rid of it. So it's important to recognize that the capsule itself doesn't always make them harmful, and it doesn't always make them resistant to your immune system entirely. In fact, it rarely does that. It just makes them a little harder to kill, and it can make a bad bacteria worse. And to prove that they're not always bad, capsule's not always bad, you actually have good bacteria in your mouth that are stuck there along with their capsule in your teeth. So you actually have good bacteria in the mouth that are not pathogenic, at least not thought to be, and they actually form little establishments in the teeth by through the formation of a capsule. So the capsule helps them stick and helps them keep in your mouth and maintain themselves as part of the natural biota in the mouth. So it's not always bad to have a capsule, but to clarify, when you have a capsule, it makes a bacteria harder to kill, and therefore that's a bad thing when you have a pathogen that might do harm to you. Last part of chapter 4 is a discussion of bacterial endospores. What an endospore is it's a life stage that some bacteria produce or go into when the environment becomes relatively harsh and the nutrient content, for example, tends to go down. One of the things that bacteria have evolved to do is to figure out how to survive in the worst of conditions, in life in general, but microorganisms at the front of the list of the most hardiest organisms that can survive almost any conditions on Earth. This is why they're found ubiquitously, right? Most things are not ubiquitous because they can't survive everywhere they go. When it comes to bacteria, there's almost some kind of bacteria that can survive any condition. There's very few exceptions to that. One example of how they're able to do this is through the formation of an endospore. So some bacteria are able to survive in really harsh and unforgiving conditions by producing an endospore. And the endospore is this really hard, protected little structure that is like a seed, really. It's very much like a seed produced by bacteria. It's this little protected little structure that contains the basics of the cell, and it allows it to survive any sort of harsh conditions. And once the conditions are favorable, that little spore, like the seed, once it gets water and once the temperature is just right, it'll sprout back to life and go back into the active stage. So essentially what we have here are bacteria that can go through two life stages. One life stage is called the vegetative stage. That's the active stage. And yes, that's kind of an ironic term. One of the students pointed that out to me, that you think of a vegetative state as like somebody who's unconscious in a coma. Fair point, as those brought to my attention. I honestly didn't even think about it. I've heard this term for so long, I've been brainwashed. But the way that it's used in this case is kind of counterintuitive, really. The vegetative stage in this context actually means active. Go figure. But that's what that actually means, metabolically active and growing. So when we talk about a bacteria in the vegetative stage, what that really just means is when they're normal, basically. Most bacteria aren't classified as being in an active or vegetative stage because that's just the way they are always. So just think of that as being normal. Like that's, in a lot of bacteria, they don't have any other stage of life they go through. They're just in a normal stage. They're either alive or they're dead. So in most cases, you don't even call it anything because it just means that they're alive. In an endospore, forming bacteria. What we really have is the active stage, and then an alternative stage where they go quote unquote dormant, or they form these dormant bodies called the endospore, which becomes what's called the endospore stage, which makes sense. So what this really is it's a stage in which the bacteria goes dormant by producing this shell around itself, and in that shell it can survive almost any condition until the conditions once again become favorable, and then the bacteria can emerge. And that's under the assumption that they will once again become favorable. That may not always happen, but if a bacteria finds itself in a favorable spot, it can then emerge and go back into the vegetative stage. So let's show some figures here. Here's a figure showing a normal bacteria in the vegetative cycle. It's also important to notice that like a normal bacteria, they will simply operate in a healthy, normal environment. They will simply operate under the normal context that other bacteria would, meaning that they will take in nutrients, make copies of themselves, and go through the life cycle as they normally would. It's only when the environment becomes intolerable that they will actually go into this alternate life cycle known as the endospore life cycle. And here what ends up happening is the cell produces a shell around the genetic material, and it goes dormant in the process of germination, in which the spore is actually formed. Now, once that spore is formed, here you can actually see the cell is still intact, but the spore forms here. Eventually what happens is the cell breaks away, and only the spore, or the endospore, is left here. Eventually, these spores will, theoretically anyway, find a suitable environment in which they can go back into the vegetative stage, re-emerging into an active cell and going back into the vegetative cycle. And if the environment once again becomes harsh, they can jump back into the endospore cycle. So it's really just a survival mechanism. It's like plants producing seeds. The seed is able to survive the winter. It's able to survive the cold and any sort of condition until it finds itself in a favorable environment. The endospore, here's some images, by the way, showing what that looks like. Here you're looking at, in this figure A, normal bacteria. Here you're starting to see them begin to form an endospore. Inside the cell, what's happening is the endospore is forming on one end, so they get this kind of tapered look to them here. From a cross-sectional view, you can see here the endospore forming on one end, and here you can see this is an endospore that is fully formed here in this part E of this figure, and you can see how that makes the cell kind of bulge on one end, as you can see down here. So that's what basically happens. It's like a seed growing within the bacteria. I call it a seed only loosely. It's not actually a seed. It's an endospore, but it's almost the same kind of concept as like a seed. Here you're looking at a picture showing a bacteria emerging from the endospore. So this is the endospore here, and this is showing when the conditions are favorable, that bacteria will literally just grow right back out of the endospore and go right back into the active or vegetative cycle again. Generally, when we say favorable, what we're talking about is water. One of the key things that will make microbes go into an endospore is a lack of water, dehydration, lack of nutrients. Typically, it's when there's a low level of nutrients. These bacteria have adapted to respond by going dormant. And when there's enough nutrients then available later, they'll come back out of it. And so it gives them an opportunity to survive no matter what the conditions are. Now, the big significance of these, besides the fact that it's kind of an interesting thing, where this really becomes very seriously significant is that endospores are literally the most difficult living organisms to kill on Earth, as it states here. Literally. When it comes to endospores, they take a significantly greater amount of care in order to exterminate their presence than really any other type of organism. And when this comes to surgical procedures, when it comes to aseptic procedures, working in a clinic where you're giving vaccinations, for example, working on people, you have a you have a harder ability of actually getting rid of the bacteria in order to make those environments sterile or aseptic. And so for this reason, endospores tend to be problematic. Where they tend to be most problematic is if you have surgical instruments that aren't properly cleaned, then they can survive and go on to infect an individual who's not or who's operated on with those surgical instruments later. Now, this is not usually a problem in the modern world because we have the proper ways of sterilizing tools and getting rid of those microbes. Um, but really, what it takes to and just put this in context, what it takes to kill these endospores consistently is something like an autoclave. And many of you may be familiar with an autoclave. If you're not, an autoclave is a machine that basically heats things up to a really high temperature and holds it there for a long time, about 20 minutes. I say a long time, about 20 minutes, holds it at a high temperature and then slowly releases that excess heat to bring it back down to normal temperature. So it's very basically just a very sophisticated heating machine that's designed to heat things up to a really, really hot temperature and then hold it there under pressure. All of that takes place under pressure. And what that does is it consistently kills all living things. But really what the point to make about that is that you really wouldn't need that if it wasn't for an endospore. Most things you can kill with hot water. If you just boiled them for a couple minutes, most things you would kill pretty easily. When it comes to the endospores, they're significantly advanced at surviving in these types of conditions compared to other organisms. So you have to really go that extra mile to kill just the endospores, whereas most other things would be killed pretty quickly and pretty easily, relatively speaking. So there's a great significance to their presence because that means you have to go through all these extra procedures, and that's why we need the autoclave. Now, from a food context, bacterial endospores are really problematic in food canning. These are common in the soil. Bacterial endospores, as I'm going to emphasize in the last slide here, they're very common. They're found in soil. Therefore, anything that you grow from the ground is going to have endospores on it. That has to be an assumption because it's virtually always true. So endospores are ubiquitous in the soil. Anything coming from the soil has them, and you really can't clean them off. You can wash the food, you can even scrub it, but you're never going to get rid of all the endospores. So when you put endospores into a can or you want to preserve them long term, what will eventually happen is those endospores will emerge in the can and go on to spoil the food if not treated properly. So the best way that they've found to deal with that is to basically sterilize the food once it goes inside of a can. So the canning process basically involves sealing the food in the can and then putting the cans, as you can see here, in this great big machine that is basically a gigantic autoclave. It's basically doing the same thing to the food that your autoclave is doing to the surgical instruments. And it's basically sterilizing all that food to kill all the endospores that are surely to be found within that food before that's done. This allows the can then to sit on the shelf at room temperature for years before it eventually becomes undesirable to eat. But without that, you would have lots and lots of rampant infection. Endospores would commonly grow inside the can, and food would either be spoiled or people would be eating it and getting sick. So the other thing I want to mention about that, and then I'll move on here, that's not good for your food. A lot of you probably know that canned food isn't that healthy, but if you don't know that, then it's a good time to learn. Canned food is not that healthy for you. It's really not the best way to get vegetables. I'm not saying that you shouldn't eat it at all. There's some benefit to it because you get fiber and things like that, but generally speaking, it's not good for you in terms of getting your vegetables. You're not really meeting your vegetable intake if you're just eating canned food. The reason for that is because the sterilization kills a lot of the nutrients. It zaps a lot of those vitamins. It zaps a lot of that good stuff in the food that you would otherwise get if it wasn't sterilized. Honestly, the best way to eat food is frozen. I don't think it personally tastes as good, but that freezing preserves a lot more of the nutrients than the canning process. In fact, the canning process arguably destroys most of the benefits of the food. There's still a handful left, but most of the benefits are actually destroyed in the canning process. So that's not only relevant to the infection and food spoilage side of it, but also from the nutritional side of it. And it's good to know that that's not really a good spot to get your vitamins and good vegetable sources. Most of you probably know that, but it's important to mention that. Last thing, I just want to kind of summarize why endospores are significant. You can be guaranteed to have this question asked in a written or matching or fill in the blank rather type format. So make sure you look at these carefully. They're not that difficult, but endospores are very problematic for the medical community in the medical profession really. for three general reasons. And one is that they're very common. They are ubiquitous in the soil. They're found in the water. They're going to be found floating through the air. They're found on people's shoes. They're found on people's bodies. They're found on everything. Um, they are very, very, very common. These are not rare. These are not found in obscure spots on Earth. They are almost as ubiquitous as any type of microbe that you'll find. So you may not find them literally everywhere, but you'll find them in most places that you look. So they're extremely common. The assumption has to be made that they're present. Anytime you're talking about sterility and aseptic procedure, you have to assume that they're there and take precautions in order to deal with them. Second, they're difficult to kill. We talked about the fact that you have to go through all these extreme heating. Bleach, of course, will kill them too, but you can't just bleach everything. So really, the most effective way is to heat things up. And then finally, we haven't talked about the examples of these. We'll talk about some of those later, and actually do a lab on this. But they are capable of infection and disease, and you might even add to that food spoilage. Infection, disease, and food spoilage. So they're able to do some damage. And if you look up endospore associated diseases, you'll find things like gangrene. You'll find intestinal infection. You can find things like botulism poisoning, anthrax, inhalation anthrax, cutaneous anthrax. These are diseases associated with endospore forming bacteria. Even tetanus is another example. So they're capable of some pretty nasty diseases, not to mention all the food spoilage that would occur from a much wider variety of species. So for those three reasons, they're very significant. They always have to be accounted for, and they always have to be assumed to be present. So when you're canning food and things like that, you have to go through the right measures to get rid of them. All right, that is the end of chapter 4. As always, let me know if you have any questions. Send me emails and write your questions down and ask in class. And I will see you next time we meet. And we will start chapter 5 on our next lesson. Talk to you later.